So if you don't have a big fancy schmancy machine like a Coulter counter, you're going to have to count your cells by hand using a tool called a Hemacytometer, and you'll just count it with a cell counter. The cell counter has its own cover slip. It costs about $8, but it's the thickness that we need to make this happen. So the very first thing we're going to do is we're going to put the cover slip on our Hemacytometer. There are these little grooves that we're going to load the cells into, and this is the area we're going to be counting our cells on. And here's the cell suspension that we're going to count. I first of all want to pipette this up and down just in case my cells have settled. And the other thing is I don't like taking from my stock to count. So I'm going to take a little tiny bit of cells and put them into a little 1.5 ml tube. This way I can keep these guys sterile, and then I can play with these. So I'm just going to grab about 30 microliters of cells from my cell tube, and I'm going to load it onto the Hemacytometer. And so I've gotten my microliters in a pipette man with a pipette tip, and I put my tip right at that groove, and I'm going to let it by capillary action fill it. And it's very important to do both sides. If we don't fill both sides up, then we don't have the right height from the cover slip to the grid. Okay, I'm going to overfill this so I can show you what it looks like. And what has happened is I've overfilled it. And we never want to overfill our Hemacytometer because then we don't get the right cellular count. And so what we're going to have to do is we're going to have to start again. So now I've gotten a nice clean prep. Both sides are filled. The cover slip was installed before, and I'm going to take it over to my microscope gently. And I'm going to put it on my microscope stage. One of the tricks here is to make sure that the light is going over the proper part of the grid here so that we can find it. So move it around until the light is actually shining through the appropriate part of the grid. And so now I've located the grid, which is just past that loading area, and the grid looks like this. That is the entire grid. The middle part is for smaller cells like red blood cells. We're not going to be counting this little tiny cross shape. We're going to count one large one large square, a second large square, a third large square, and a fourth large square. So what I'm going to do now is I'm going to come and find and focus on this square. So this is what a large square looks like, and what I'm really looking for is this really very delineated line here and this delineated line here, and this is my large square. It is separated into 16 smaller squares, 4 rows of 4. So to help the counter along, I'm going to make sure I get these 16 squares in frame. And so what I'm going to do now is I'm going to grab my little counter, and every time I see a cell, I'm going to press it. So a cell is this little tiny dot here, and I have 2 in this little tiny mini cube. So you can see how it would be hard to keep track if I didn't have some lines. So I'm just going to go about the business of counting this entire cube. So and I got about 49 cells. I do want to avoid any cells that are outside of my grid. And now I have my first count, and I'm going to move over to the next cube, the next large square. So now we can appreciate that the really very segmented portions on the left, and then I have one on the bottom, so I've moved over to the right, and then I will count these cells. And then this is my fancy notebook over here, but I'm gonna write those 4 numbers down. So I got 58 this time. So I'm going to write that down. And I'm gonna continue doing that till I've done all 4 of my large squares. And so now I have counts for all 4 large squares, and what I need to do is I need to take care of my hemacytometer and clean it and put it away.